Hello, everyone. I'm excited to be here today and present our recent work at ISMB ECCB 2021. Special thanks to the ISMB and EvolCompGen program committees for this wonderful opportunity. I'm Janani Ravi, a computational biologist at Michigan State University. Before I start, I would like to give a shout out to the awesome undergraduates, Sam, Lauren, and Joe, who've led this project in style. This is still work in progress, and we would love to receive your feedback during our live Q&A. For those of you who are unfamiliar with what we do in our group, we work on both the pathogen biology itself and the host response to pathogens. We develop computational approaches to study molecular mechanisms of pathogenesis and host-directed therapeutics against infectious diseases. And on the pathogen side, we develop approaches spanning several different scales: proteins, genomes and genomic features, and small RNA discovery. We have also picked up a few favorite pathogens and infectious diseases during the past few years, thanks to some wonderful collaborators. In our group, we are interested in understanding several aspects of what makes each pathogen tick through the lens of computational evolutionary biology. Most of us working on pathogens are interested in fundamental questions such as pathogenicity, host specificity, environmental and pathogenic adaptation, and the evolution of antibiotic resistance more recently. If not fundamental biology, we are always thinking of different ways to squash the bug. Most of these fundamental and translational questions, in great part, can be answered by studying the conservation versus unique molecular features and specificity versus sensitivity. We build streamlined approaches for molecular evolution and phylogeny at the protein level and comparative pathogenomics and pangenomics at the genomic level and gene level features. And today I will focus on the former. I first started working on using the molecular evolution approach to study a pan-bacterial stress response system during my postdoc at Marla Genero and our collaborators at NIH with Arvind and Vivek. Since then, the approach has been generalized and now tested by us and others to better understand the nature and evolution of proteins and protein families. This generalized approach is what I will be focusing on today, along with the recent web applications that we built since my last talk at ISMB. This new approach that I'll be showing you were built by my undergrads, Sam, Lauren, and more recently, Joe. They have worked with me on streamlining this approach to build a user-friendly web application for biologists and R package for computational biologists. So before I take you through the specific steps and the details, also using demos in the web applications, let me highlight the main steps here. Our approach starts with query proteins of interest in different shapes and forms, identification of a comprehensive set of homologs, characterizing both the query proteins as well as every one of their homologs in terms of their domain architectures, and for bacterial proteins, also in terms of their genomic contexts. And finally, studying all of these homologs and protein families in the context of evolution. I will demonstrate the power of our approach by using the Phage shock stress response system as an example. I will alternate throughout the talk by giving some context first and then showing you a quick demo of how this works in our web applications. The PSP system is critical for bacterial envelope cell surface, stress response, and membrane stability and integrity. So it's basically very important. Now, while the PSP stress response is highly conserved across phyla, there is a lot of diversity, as you can see, that are highly lineage specific. There are a lot of shapes and colors and protein names here. Don't worry about any of that. The main thing to focus on is that there is only one namesake protein, the black one, the PSPA, which is actually conserved across lineages, while all the other interacting cognate partners are actually very different. Now, the colors are indicating something. For instance, all of the orange ones are transmembrane and the teal ones are all transcription regulators. To say that, it doesn't matter which exact transmembrane region it comes in, proteins it comes together with, as long as it has transmembrane proteins and transcription regulators for the stress response system to function fully in these different lineages. So we got very interested in what these different partners are, how the lineage spread is, and what this means for the biology. So we started a very in-depth study in terms of that. So first, when I say we look at the homologs across the tree of life, we do a few different things in addition to just routine blast search. And what I mean is we start with multiple starting points, which is at least 8 to 10 across distinct lineages. And we do iterative searches as well to pick up remote homologs. And finally, we also do domain-centric searches in addition to full protein searches for PSPA, as well as every one of its cognate partners to pick up all of the varieties that we have in these PSP systems. So let me now take you to the PSP app, which was actually built by my undergraduate Sam Chen here at Michigan State University. So the first thing I'm showing you is basically we have the PSPA protein, all the other cognate proteins. This is dynamic, searchable, queryable, interactive web application. You can go to the NCBI protein here. We have also shown the domain architecture, the genomic context for every one of these homologs. We can see the query heat map, which is PSPA, as well as all of its cognate partners and their lineage spread across the tree of life. That's the very first thing that we look at, which is where are the homologs and who are these different homologs? What are their specific traits? Okay, now I'm going to go back to my slides. So we reconstruct the domain architectures by characterizing the protein and every one of its homologs. And we do this in a few different ways, using sequence alignment and clustering algorithms to identify homologous groups and protein families. We do profile matching using protein domain and orthology databases, prediction algorithms for signal peptides, transmembrane regions, cellular localization, and secondary and tertiary structure. So all of this to finally get at what this domain architecture really is. And what I mean by that is the order in which these different domains are coming together in a given protein. And this is an example of a sulfur acquisition system that we studied with Neil Hammer's group at MSU. And we are studying how this system has evolved, not just in Staphylococcus pathogens, but also across gram-positive bacteria. And this figure is showing us what the domain architecture is by looking at the different kinds of analysis. We've also used it to study the S-layer proteins that are very important cell surface armors that are important for pathogenicity and virulence of Bacillus anthracis. And we looked at this across gram-positive pathogens in a recent publication with Antonella Fioravanti. So we can infer which domains are most likely to interact through what we construct to be a proximity network. Because especially when you're looking at domain architectures across thousands and thousands of homologs, we need a reasonable way to summarize our results. And this is one good way to do it. For instance, what I'm showing here is, let's say about 140 homologs, right? And most of them come in this configuration with domain 1 and domain 2. So when you look at it in the network, domain 2 is the most popular one. So that's a pretty big node here. It's connected to these other domains, and the size of these other domains depends on their frequency, and so does the edge weight. So now let's look at these domain architectures in the PSP app. So here again, as I mentioned, we can do this for PSPA, or we can do it for a slightly more interesting domain that actually shows a lot of variety in terms of the other partner domains that it comes together with. So we first summarize the domain architectures that we find across homologs. We can see which are the prominent lineages that you see a given domain architecture in. So it occurs by itself pretty much everywhere in all possible lineages. But some of these other ones are highly lineage specific. So you only see it come together with PSPC only in actinobacteria. Then we construct something called upset plots, which are basically fancy ways of doing set diagrams when you have multiple interacting sets. In this case, we are looking at where these different domains are coming together to form these domain architectures. Again, as I mentioned, this app is interactive, so you can explore it a few different ways. And finally, we can actually summarize by looking at these different combinations of domains and co-occurring domains with respect to our main protein and domains of interest. And in fact, if we want to look at the whole PSP stress response network, we can do that too, because now we have all of the data here. It seems like it's disconnected, but now once we start exploring all of the PSP proteins, their cognate neighbors, actually a lot of them are connected to each other. And we discovered a much larger PSP stress response network in not just bacteria, but also in several archaeal and eukaryotic lineages. So with that, I'm going to go back to my slides. So these are a few different ways in which we summarized and identified novel domain architectures in the PSP stress response network. And finally, for bacterial proteins, as many of you would know, looking at proteins and their domain architectures alone is not sufficient to inform function. We also have to look at the genomic context, meaning who are the neighboring proteins? Are they part of an operon? Are these co-transcribed? Because that is also extremely important in defining function of these bacterial proteins. So what we next do is basically to look at the neighbors of these different homologs. And we characterize this in our web application as well. So here I'm now going back to my app under the genomic context tab. And again, as always, we do this for every one of our PSP cognate proteins. Again, we have a summary table. For instance, we see that this context with ABC is only coming together in proteobacteria, whereas this is the one that we see in actinobacteria and Mycobacterium tuberculosis that we discovered in my postdoctoral group. So you can see what are the different genomic contexts, and already we have identified several novel contexts that weren't described before in the context of the PSP stress response system. We'll look at the different combinations in which these domains come together. Now that's too crowded, so we can probably look at the primary ones here. And we identified several new domains that are coming together, co-occurring with the PSP stress response network. And finally, we are going back to look at all of this in the context of phylogeny. So as I mentioned, we identified several novel genomic contexts for PSP. So this is just me giving you quick snapshots of what we discovered. And finally, because we have homologs now and the characterization of these homologs across the tree of life, we go back to look at these in terms of their phyletic spreads. For this, we have dynamic sunburst plots. If you want to increase the lineage, for instance, you can see that these are also in gamma proteobacteria more specifically rather than just proteobacteria. We have the phylogenetic tree for each and every one of these proteins, and these are colored by the lineage. We have the multiple sequence alignment of these different critical PSP proteins. And more importantly, and this is especially useful for the PSP system, is we actually also have the paralogs. The nice thing about looking at these paralogs is now when you click on these, you can also see what kind of paralogs these are. Meaning, are they basically part of the same context? So was it more likely to be a case of gene duplication? Or are they a result of horizontal gene transfer? And you can see this by comparing homologs from across lineages and placing them on the tree. So we can do all of this analysis again for every one of these proteins. And this app is already pre-populated with the data. So this is the PSP phylogenetic tree that we constructed. And we actually discovered that taken together the PSPA and the SNF7 escort system from eukaryotes, the superfamily dates all the way back to the last universal common ancestor, which was pretty cool. If you want to dig deeper and go into one particular lineage, we can also do that. And we did that for actinobacterial PSPAs and actually discern some of these specific cases of gene duplication versus horizontal gene transfer from firmicutes. And we also applied variations of this approach to discover the phage defense systems in Vibrio cholerae with Chris Waters' group, and this one is currently under review in Cell. So in summary, with our very detailed computational evolutionary approach, we traced the PSP SNF7 system back to Luca. We discovered the larger stress response network. We identified several new themes and 15 plus novel partners and the distribution of these across phyla. And because of this detailed analysis, we were also able to predict some novel biology and what this means for membrane stability and homeostasis, which is at the root of the PSP stress response function. So the PSP web app is just one particular instance where we have collated all of our results here. We had too many results and too much data, as I mentioned, and we just didn't know how to reconcile them. And that's when the first version of this web app was born. And Sam, the undergrad who was then a sophomore, he just came in, and within 3 to 4 weeks, we had a prototype, which was just beautiful. And then, as always, the app evolved and evolved and evolved. And now this is what we have with the entire PSP data that's been populated there. So we keep adding additional features through and through. We use it to search through our results, create visualizations on the go, and to also change thresholds and other kind of layouts. These are just basically aesthetics that we added more recently. So the practical challenges that we realized when we were building the approach on the backend itself is that a lot of these current tools can only leverage subsets of these data to hint at different facets of proteins, bacterial proteins, and genomes. So these are all present in siloed servers and databases. And it requires a lot of knowledge about the tool installation, how to manage a server, how to do all of this data analysis and visualization. So it requires a niche kind of expertise and several years of development of this method. Because, for instance, you do know that there are a lot of these independent tools, right? Similarity searches, BLAST has been doing a fantastic job for several decades now, and more recently, MMSeq does a much faster job of it, right? And then phylogenetic spreads, there are several tools that many of you know for building phylogenetic trees, multiple sequence alignments. And then now there are also some really cool tools like InterProScan and other ones to study domain architectures. So all of these do exist, but there are still no such unified frameworks and tools to perform all of these analysis under one umbrella. So that is when we decided, okay, this was really cool and really powerful, not just for PSP, but also for a few other projects that we have worked on. So why don't we generalize this computational evolutionary approach so that we can use it for any gene and protein of interest and any set of genomes of interest? So the result is that we have now built a prototype of web application for biologists, and we are also converting the backend, all of our scripts, to release as an R package sometime this fall for computational biologists. And we're really excited about this. A few of our favorite pathogens for the past couple of years have been Staphylococci, Mycobacteria, Vibrio, Bacillus anthracis, and Brucella. So we have tested several parts of our approach on these different pathogens and really cool questions there. And I'll be showing you that in a little bit. Okay, so what does the current version of the app now have? So we can do protein family construction. You can start with a query protein of interest or a comprehensive set of homologs. If not, we will get the comprehensive set of homologs for you. If you have done analysis elsewhere from, say, the aforementioned BLAST or InterProScan, sure, you can bring in those results too into our web application. If you're starting with just protein sequences or sequences of your homologs or accession numbers, we will allow you to do the domain architecture, the detailed domain architecture analysis within our web application. If you're starting with BLAST results or InterProScan results, you can also do the phylogenetic analysis here, or you can do all of the above. So these are the main things that you can currently do within the web application. So MolEvolvR beta is now here. We would love for you to test it and give us feedback because we are hoping to submit it really soon, sometime in July. So please use the live Q&A, post your questions, tell us what are the tools you would like to see in the app. If you would like to start a collaboration, we are interested in hearing all about this. So let's get started. Now I'll take you to the new app. So this is how the app looks, and I'm hoping that it's been loaded. Okay, and what you can do is basically start your analysis, which takes you to the upload tab. So here, as I mentioned, you can start in a few different ways. If you just want to start with accession numbers, you can start there. You can generate FASTA sequences, or you can upload FASTA sequences, generate multiple sequence alignment, and then proceed with different combinations of these analysis. And as I mentioned, you can either do full protein or split into domains and do domain-centric analysis and then collate your results. If what you're entering are already homologs, you might want to indicate that and directly proceed to phylogenetic analysis. So you basically can customize your analysis here. And if you've already run a BLAST search elsewhere, you can bring that in. So this is an example BLAST output data that I'm uploading here. And this is how a typical InterProScan data file looks. So you can bring in all of this and do the corresponding analysis here. Or basically, if you have submitted any of this, you're going to get a 6-letter code, and you can use one of these codes, and you can basically retrieve the data here. And at any point, if you want to know what needs to be done, or if you're not sure which format, how to bring it from the other application into here, we have a detailed help documentation page here. So feel free to explore it. Okay. So now let's say I'm just retrieving an example code here from a prior analysis. And since this is running on our backend, if it's a very detailed analysis, it might take 2 to 3 hours, but you can always come back and retrieve your results here. Okay, now while it does that, oh, it's done, cool. So this is a quick summary of the different kinds of analysis that have been done. Because this is full analysis, it will have your domain architecture, phylogeny, and all of that. So you can actually explore it even here. But let's go to the corresponding tabs. So first, let's look at data. So again, this is very similar to the PSP app, except that the really cool thing is this is with your data. If you want to add other domain architecture columns here or any other pieces of information, feel free to go ahead and add that here. Okay, you can download your data. You can search for specific ones here. If, let's say, you're only interested in what's happening in actinobacteria, because we've added the lineage information, you can now select that there. These are your input FASTA sequences. And since I'm starting with at least 3, now if I want to restrict my search by only those query proteins, I can also do that. Okay. Now from here, for instance, even from the results summary tab, I can take you to domain architecture. So if I go to say 100%, this particular set of proteins that I'm looking at seems to have mostly this COBW domain. And that's because I'm selecting PFAM. Phobias gives me my localization, whether it has a signal peptide, whether it's cytoplasmic, non-cytoplasmic. Gene 3D is from the cat. It basically gives me 3-dimensional structural domains predictions. This is TMHMM prediction. This is signal P. This is a disorder prediction. Smart PFAM, superfamily, tigerfam. If any results appear in any of these analyses, you're going to see that here. If not, basically, it's not even going to appear here. Okay, so you can basically explore your results here, again, for each and every one of your proteins. You can look at the heat map of your protein of interest. And you're seeing that this is primarily a proteobacterial domain, but you also see a lot of it in actinobacteria. So this is just a quick snapshot. And you can export these images. If you want to change the theme, you can explore and download these. You can generate the domain proximity networks that I showed here. Okay, now you just have 2, so there's not much happening in the way of domain proximity network. But in theory, okay, so if I'm looking at 100% of the data, it gives me a little more information about all the interacting domains. Again, there are a few different layouts that you can check out. You have the upset plot to see not just which are the domains, but which are the combinations of these domains that co-occur within a protein, and what's the frequency of this co-occurrence. And as we saw, COBW by itself was the most frequent. So these are just different ways of summarization and visualization that we show you within the app. And finally, we also because we can do the interpro scan analysis in the backend, we also add these additional visualizations for you. And these are picking key representatives from different lineages. And I can add as many as I want here, or if I want to look at these analysis, but by protein instead, I can basically flip this around. And again, you can save all of these plots. So now you see, basically, these are two different analysis, but basically it's matching against the same part of the protein. Whereas if I look at Phobius, it seems like it has an additional prediction here to say that this is cytoplasmic. Okay. And finally, we can look at the phylogenetic analysis. And again, you may remember all of this from the PSP app, but we are doing this now with the proteins of our interest. These are the starting proteins. Here we are starting with just 3, so it gives us a quick snapshot. Let's see if we can increase this a little bit. 5 is too little. And then it dynamically generates these trees. And you can pick representatives by lineage or by species, depending on how you want to construct this tree. And it does all of this filtering based on the backend data that it has already generated. So this is the phylogenetic tree overlaid with the multiple sequence alignment and these different colors of the different amino acids. And finally, if we are really lucky, we can also look at the multiple sequence alignment for these proteins. We are very lucky today. Okay, so we also have the multiple sequence alignment, which you can download as a PDF. And as I mentioned, everything in this app is dynamic and interactive. And we are working on making this as smooth as possible and adding a little more documentation and hopefully a few tutorial videos as well to help you navigate the web app. So please do check it out. And we would love feedback during the Q&A, as I mentioned. So hopefully it'll be on bioRxiv soon before the end of the month. And with that, I'm going to go back to the last part of my presentation. So eventually we're going to be adding genomic context as well and a few other analyses like MMSeq, RPS-BLAST, and a few other ways of analyzing these proteins, not just within bacteria, but basically, as I showed you, across the tree of life. So we have demonstrated different parts of this approach in great detail in a few different systems, not just the PSP ones that I showed you initially, but also with Chris Waters' group from MSU, Anto Fioravanti from Brussels, and with Neil Hammer, where we are studying staphylococcal pathogenesis. And now we are getting really close to finishing the web application, hopefully the R package as well. Oops. So with that, let me thank the team. They have done a fantastic job. I'm really, really lucky to have worked with these super smart undergraduates at MSU. And we've gone through the usual routine of knowing we have figured everything out. And then 2 years in figuring out that we are nowhere close. And finally, now we are very close to the end and we are super excited once again. And John has basically set up the backend server, and without him, we will not have this web application. This is the awesome group of undergraduates that I'm very lucky to work with. A few of them are also giving posters and talks at ISMB. I've also had the good fortune of working with fantastic collaborators, both at MSU and elsewhere, including my past collaborators at NIH and Rutgers. Many thanks to them, because that in some shape and form did kickstart this project in a very big way. And I would like to acknowledge the fantastic communities that made this work possible. So I'm part of, I founded and I'm co-organizer of Our Lady's East Lansing and Women Plus Data Science, and these communities have greatly helped our students in being an essential part of these R and data science communities. On our open site, is going to basically help with the peer review process for actually going through our package code in great detail before we submit it to our journal. Also, the work was funded by several different funding sources, both from MSU and NIH. And I would also once again like to thank ISMB and EvolCompGen for giving us this opportunity to present our work here. Please do check out our other student talks and posters. Kevin is going to be talking about our recent publication briefings in bioinformatics on reconciling multiple connectivity scores for drug repurposing or transmit. And there are two really interesting posters that we are working on right now. So this is very current. And these are both from EvolCompGen. So check them out. And with that, I'm very happy to be taking questions. I think we have quite a bit of time today for some live discussion. So let's get that started. And please share your written feedback. If you want to get in touch with us or you want to start a collaboration, we would love to hear from you. So thank you so much once again, EvolCompGen folks. And I thank the team for this wonderful work. Thank you.